of this COMSTECH NTD network. Let me tell you that series both COMSTECH and the NTD net research capacity to find new and better therapeutic solutions for neglected tropical diseases. With this, I would like to invite Professor Mohammad Iqbal Chaudhary, Coordinator General COMSTECH and Director ICCPS, to introduce our distinguished speaker.
Bismillahirrahmanirrahim. Assalamu alaikum. Good morning and good afternoon, ladies and gentlemen. You know, Patrick, we already have a house full, almost like 100 people. And I have been told that they're going to be about double of this number. So your lecture is already very popular. And this is certainly what Samar has mentioned, that through this lecture series of COMSTECH NTD Distinguished Scholars presentation, what we are trying to do is to bring science to the regions where we have COMSTECH least developed countries. And these are the areas in which we have mostly these tropical diseases. So certainly this lecture series carries a lot of importance and relevance. And the speakers selected for this lecture series are among the best in the world in their own chosen disciplines. COMSTECH is an organization which is one of the strongest organization of Islamic cooperation, OIC, 57 member states. This is the second largest political bloc, but of course we do lots of other things. And COMSTECH, which is a ministerial standing committee for science and technology cooperation, is responsible of developing science technology cooperation among the 57 member states. And that include states from South America, Suriname and Guyana, to Africa, Asia, and Europe. 57 member states spread from the west of Pakistan, which is about 7 hours behind us, and to east of Pakistan, which is 3 hours ahead of us. So there is a whole spectrum of time zones, but at the same time, the scientific capacity of these countries are also very dissymmetrical. So we have some very good research centers, some of the finest research centers, including the one which we have in Karachi, which you have very kindly visited. At the same time, there are countries where we need to build and strengthen the science and technology capacity, and for which I'm extremely grateful to NTD Network and all of you for extending help in, at least at this point of time, virtually developing capacity and appreciation of this very important area. I am extremely pleased to introduce Patrick, who's a dear friend and the one who has visited our center. And certainly because of his effort, we were then actively engaged in this NTD network, and certainly an experience in which we have greatly enriched and learned. Patrick Steele undertook his undergraduate and postgraduate training with some very leading names. Professor Jim Thomas at the University of Oxford. Following a NATO CERC postdoctoral fellowship, he was with Professor Gilbert Stock and the renowned organic chemists, some of the finest chemists with whom he has benefited. Gilbert Stock was from Columbia University, New York. He joined the staff at Durham University, where he is currently a professor of organic chemistry and chemical biology. His group studied problems in organic synthesis, iridium and copper catalyzed correlation and methodologies, and chemical biology with particular interest in neglected tropical diseases. So he has actually combined his in-depth understanding of chemistry with his interest in biology, and he focused on tropical diseases, notably leishmaniasis, and also plant chemical biology, plant hormonal signaling, and herbicide resistance. He is currently the head of organic chemistry section at Durham University and was an elected council member of the Royal Society of Chemistry, Chemistry Biology Interface Division 2013 and 2019. This introduction of Patrick is certainly an extremely brief version of what he has done, and he is certainly among the top organic chemists of the world with a global vision, because he traveled a lot, and we are looking forward to have him in Pakistan. His visit is long overdue. Now, with my presence in Islamabad, we are so eagerly looking forward to welcome him in Pakistan, both Islamabad and Karachi. Patrick is going to talk about identifying and validating new drug targets for neglected tropical diseases. So is in fact a blend of very interesting chemistry as well as biology, and for that we are eagerly looking forward to have the lecture of Patrick. Patrick, over to you now.
Yes, thank you very much, Iqbal, for that very, very generous, almost embarrassing introduction. I thank you again to Dr. Samar and the organizing committee for giving me the chance to present. So let me just get my screen up and running. Hopefully people can see that okay. Okay, so again, thank you to the organizing committee to be the latest speaker in this series. It's been a great lesson for me to come and learn some of the science other people are doing. I'm terrible for timing, particularly when I speak online. So let me start by acknowledging that what I'm going to talk today it's been a team effort, it's been a collaboration, it's been an international collaboration. I have my two fellow leaders, there's Paul Denny at Durham and Bartira Rossi Bergman at the Federal University of Rio de Janeiro. And they have taught me what little I know about parasitology. What I get wrong is my fault, I'm a poor student. And then most importantly, it's the people in blue. These are the people certainly who've passed through my group at Durham who've done the work I'm going to show you. There'll be others in Brazil in particular who've also contributed. We've been supported by people over here on the right, particularly highlight Emka Pol, who keeps me straight on structural biology. These other people have helped us with key experiments along the way. And of course, we can't do it without support from various people, particularly the NTD network, the Royal Society and CMPQ. So we'd like to thank them for all their support. And in case I hope to repeat this sort of thank you at the end. Okay. Neglected tropical diseases, they're a collection of 18 to 19 diseases which are not well represented in the modern pharmacopoeia. They're global diseases, they're about 1.4 billion individuals are probably affected. That's about a sixth of the world's population will have one of those neglected tropical diseases. They're largely a disease of poverty, so it's the poorest people in the world who suffer them, and they have a major socioeconomic impact that's probably best measured in this term of disability-adjusted life years. And this is a very old slide, and we look at the spending per disability-adjusted life years. Those 20 neglected tropical diseases are right at the bottom of the league table down here when we compare to other things. And obviously with recent things affecting the world with the pandemic, that situation is only going to get worse. So we need research and we need development in this area. Okay, so that's the political message. Let's go on with the science. As Iqbal said, I'm particularly interested in leishmaniasis, disease caused by the parasite leishmania, or the leishmania species. Here are the statistics. It's a growing problem. It's endemic across the globe, particularly in South America, Sub-Saharan Africa, and South Asia. There are lots of people infected. There are more people at risk. There are many new cases of leishmaniasis, mainly cutaneous. The numbers of deaths are probably diminishing, and that's largely due to the efforts in India and related countries to do the eradication program for visual leishmaniasis. The big problem is there's still a huge number of disability-adjusted life years, so it's one of the most serious of the global health problems caused by neglected diseases. Okay, I don't know how many, if everyone on the call is an expert, so very brief, very quick introduction to the life cycle of the leishmania parasite. We start in the insect form, this flagellated promastigote, and that lives in the midgut of the sandfly vector. When the sandfly takes a blood meal, the parasite or the metacyclic form is taken up in the bloodstream and phagocytized by the macrophage, where it transforms into these amastogotes. And this is the stage where we see the disease, the infective form of the disease. The disease comes in several forms. There's the cutaneous form. We get these open lesions on the skin. This is the most common form of disease. It's probably the most neglected of the neglected diseases, because people assume because it's self-healing, we don't need to worry about it. Or that's not, maybe a bit of a short statement, but it's less considered, less important. The most disfiguring is the mucocutaneous form, where it destroys the mucosal membranes and the lips and the nose. And then there's the visceral form, which is fatal if not treated. And this is where the problems in leishmaniasis really come. The treatments that we currently have aren't perfect, but it works in. We still use antimonials, so pantastam and glucantine were developed in the 1920s and 1930s. They're still used, they're toxic. And that sort of deficiency encompasses all the drugs on the screen. These 5 over this side are all given intravenously. Some of them require multiple daily injections. Miltefosine is the only oral treatment. It's licensed for use in India for visceral leishmaniasis. It shows mixed effect when you test it in other species of leishmania across the globe. So basically there's a problem there. So there's a challenge to developing new drugs. And because of the population that can't afford drugs, that's a challenge for all concerned. So the pharmaceutical industry struggles. I should just say, there is compounds coming through from public-private partnerships which look very exciting. Whether they get to that approved drug remains to be seen. The challenges for drug discovery is it's a long process, 10 to 15 years. It costs a lot of money that requires a return. So as academics, what can we do maybe to contribute to this effort? We largely focus ourselves at the top end of the thing, of this sort of pipeline. Can we help with target identification, target validation? We can de-risk some projects. Maybe other people could pick them up and run them and get them through this process. What I want to do today is show you two stories of how we've done that. And if we think about that, there are 2 strategies we could follow for this early stage drug discovery. There's a target-based approach, where you start with some sort of biological hypothesis, a biological pathway that you think that if we inhibit that, it may affect the progression of the disease. You develop a biochemical assay and you find compounds that work in that biochemical assay. And then you have to prove that they work in both an animal and in the parasite and then the animal. The other way around is to go from a compound that has the observed effect in some sort of disease model, a phenotypic-based approach. And you can identify that from some sort of phenotypic assay. And that again gives you a compound. But then you have to go and link that compound to a target if you really want to be efficiently develop that into a suitable drug. So again, I'm going to show you 2 stories. One comes from each of these columns. The first one I'm going to start on is a phenotypic-based approach. This is a project that started in Brazil. It started with Bartira's group. We've shown some years ago that you take an extract from this plant you get from the Amazonian forest. I believe it's used there as a remedy for cutaneous leishmaniasis. This compound here, you can extract this chalcone, which shows some activity against the promastogote, the insect form, the intramacrophage amastogote form, and has some selectivity when you compare it against the human host macrophage. Not particularly active, but it was an active compound that showed some activity against leishmania. Now, number of people in the audience will say, well, chalcones, are they really suitable as drug leads? If you look in the literature, you can find a chalcone that's anti almost everything. It's anti-inflammatories, antivirals, antibacterials, anti-cancer, antiseptics, anti-HIV, etc., including anti-leishmanial. So the first anti-leishmanial chalcone is licochalcone A, which was reported 25 years ago and showed to have effects. It causes mitochondrial alteration to parasite, inhibits its enzyme, fumarate reductase. So chalcones are well established. Are they ever going to be a drug lead? I'm not so sure, but they can be useful chemical tools maybe to help us identify targets. And that's what I want to show you today. So natural products were established as drug leads. In fact, my colleague Paul showed you some examples of that, and we came to the Nobel Prize a few years ago. Okay, so what Bartira did, they did a little bit of chemistry in Brazil, and they were able to make an optimized form of it. So they made this slightly modified extra-methyl group and introduced the nitro group. What you see is slightly higher activity and a better selectivity index. More importantly, Bartier was able to show that this chalcone was effective against the 4 major strains of Leishmania that caused disease in Brazil. And importantly, when you compare this compound here, the CH8, with pentastan, one of the antimonials, it showed a similar sort of effect in vivo. So this chalcone does actually work. Okay, so Bartier did some preliminary mode of action studies, which have shown actually this doesn't inhibit fumarate reductase, doesn't affect cysteine protease, doesn't affect trypanothionein reductase. It might have some effect on lipid biosynthesis. What it did do was it showed some effect on the mitochondria, the size, led to some DNA fragmentation, and particularly led to upregulated ROS, which probably contributed to parasite death. But optimizing it was challenged, partly because it was quite difficult for Bartier to make, and we had no idea of the target. And that's when Bartier and I started to talk. And I guess what I'm going to say is international collaboration is good. International collaboration is good and fun. So, since this was a challenge, so we said, oh, we can make it, it's easy. And so we tried using the reported procedure, and we had the same problems. When you try and do this aldol condensation dehydration of an aldehyde with this acetophenone, you get the chalcone, and you get a lot of the flavone caused by cyclization of the hydroxy group onto the end of the double bond. We were able to address that very simply by changing around the steps. We start now with effectively a protecting group. We make this trimethylated acetophenone, we do the aldol reaction, and then remove the methyl group with boron tribromide, and we get about an 80% yield of the chalcone. So we can make this in multi-gram quantities, and we're able to do that for Bartier and give us some. We made this, we tested this compound, and again, this shows similar activity to the compound, potentially a slightly higher selectivity index because of slightly better intermacrophage amastigote activity. But we still had no idea of what the target was, and that became the challenge. Sorry, well, I should have just said this shows activity in here, it works in vivo too. Okay, we had no idea what the activity is, but you speculate. It's a chalcone, it's an electrophile, it's probably reacting with some sort of biological nucleophile, the most likely one being a cysteine type thiol. So we speculated that was the case. And so to test that hypothesis, we simply prepared the compound that lacks the double bond by alkylation of the ketone. It wasn't very efficient. I've lost you, it's only about 20%, but we make enough of this molecule to test. So here we have the compound, our lead compound, NAT22, by the student who made it, and this dehydro analog here. What you can see very quickly is this compound retains the activity against all the strains of parasite we test. This one is largely inactive. We'll see these two compounds coming back and through. This is going to be a sort of negative control that we're going to use through the assay. So what is the target? Well, we thought, okay, if we can make this compound here, where we put a tag on the end of the alkyne, we can feed it to a parasite. Here's a chemist's view of what a parasite looks like. If we're right, and there's some sort of enzyme nucleophile that's going to be alkylate or be alkylated by the chalcone, we can stick our chalcone onto this enzyme, we can lyse the cell, and we can use pull down chemistry where we use a copper catalyzed reaction to link an affinity agent to that chalcone, and we can fractionate and hopefully enrich the protein. So the first thing we need to do is to make these tool compounds. That's quite straightforward. So this is the Iridium catalyzed chemistry that we develop in our group, or we use in our group to explore CH functionalization. So we can do a CH functionalization where we selectively activate this carbon-hydrogen bond here. We can then oxidize the boron atom we introduced there to give us this phenol, and we can alkylate to introduce this alkyne tag, or this tether, onto the chalcone. And then we can do our aldol condensation as before, and we get this variant of the chalcone, the bioactive chalcone we get here. Now this retains activity against the pro-mastigote. It has picked up toxicity against the macrophage, so we're going to do all our experiments in the pro-mastigote, and we're going to make the assumption that the same target is existing and active in the amastigote. And then we make various different tags. So this one here we're going to use also has a fluorophore on it to enable us to detect things. Okay, so we have our chemical tools. Let's go to work. So the first thing that Douglas did when he did this was he simply put the compound in the parasite here, and then we cross-couple it, we link it to that fluorescent molecule here. And what you can see is this compound appears to be going throughout the cytoplasm of the parasite, potentially something into the mitochondria. If we label with Hurst, this labels the nucleus, we don't see any co-localization. So it goes into there. So we seem to have some sort of global idea that's in the parasite, that compound's getting on board and we can visualize it. If we take the parasite and we lyse it, and we do that, run them up the gel and we look for fluorescence, we can see that fluorescent tag begin to label certain two bands. The two strongest bands that appear to come out were this one at 22 kilodaltons and one at 35 kilodaltons. Are they really associated with the activity of the chalcone? How do we test that? The way you test that is to go through a very simple workflow, a competitive flow. So firstly, we treat the parent compound or the parent drug to the parasite, incubate that for a little while. We then add our probe, we then add the fluorescent agent, do the lysate, and we run the gel out again, and we look to see what happens as we increase the concentration of the drug. So one is the drug, and as we increase the concentration of the drug, you see that fluorescence decreasing for the 22 kilodalton band. We didn't see any effect on the 35 kilodalton band. So this suggested this lower band here, the protein or the proteins causing this band here were responsible or were linked to the activity or the binding of the chalcone into the parasite. So we're going to focus our efforts on this one here. We haven't done any work on this. This may be a second target of the chalcone. Okay, so you can take that band and you can take the beads and you can do either on gel, and you can cut the band out of the gel, mass spec, trypsin digest and examine the peptides, but you can simply do that off the pull down beads using the streptavidin linked beads. We do that, and you end up with the peptides, and you can sequence the peptides and you compare it against the database, and it appeared to suggest that this protein we're identifying was this enzyme here, cytosolic paradoxin peroxidase. It's quite highly abundant. That might account for it, and it's found in the cytoplasm. So there we are. Can we verify that? Well, fortunately, there's an antibody to cytosolic to paradoxin peroxidase, and Professor Angela Cruz at University of Sao Paulo was able to give us a sample of antibodies. So I'll do the Western blot, and there we are, very clear labeling of the same protein band from that there, and actually see a small trace of the dimer, and that becomes significant when we look at the structure or the reported structure for this protein in a minute. And we can tell it's a single band. We did the DIG, and we get much better resolution, and it looks like being a single protein. So we're confident that protein is what we think it is. This protein is well known, it's been well established, it's a key element of the process that maintains the trypanothione population that helps the parasite deal with reactive oxygen stress and a variety of other forms. There's some work done from an Italian group in 2012. They reported the structure of the cytosolic form. It occurs as a pentamer of dimers and exists in, so that's the dimer there, and that probably accounts for that higher band we see. I should just notice there's a cytosolic form and there's also a mitochondrial form. This is very highly abundant, so maybe we just can't detect that one in our assays. Again, it's something we're still looking at. Okay, so we were able to get the sequence of that protein, and with some help from MCOPOL, we could prepare recombinant samples of cytosolic paradoxin peroxidase. Here you can see the mass spectrum of it. If we incubate that sample with the chalcone, we see the shift in the mass spec peak corresponding to the addition of one chalcone per protein. If we take the dehydro compound, we see no labeling. So that enome appears to be important for that labeling event that we're seeing. And that probably accounts for the pull down. If we do that same competition experiment we saw before, we pre-incubate with the chalcone, and then we add in that alkyne linked probe and add the fluorescent. Again, we see a concentration dependent decrease in fluorescence, suggesting that competition is again associated with binding of cytosolic TX-NPX. And finally, in a sort of assay, we do thermal shift assay. We look at the protein stability when we titrate in the drugs. If we titrate in the enone form, we see a decrease in the TM for the protein. We seem to be destabilizing maybe that protein dimer or conformation. This dihydro form has no effect. So now we appear to have a molecule, a potential target for the chalcones with a molecular basis. Is that really associated with importance in the activity? And I take absolutely no credit at all for this slide. This was Douglas. He learned it from Jeremy Mottram, so it's testimony to the network's training and supervision by Paul and Bartiera. And I think Anna Paula and UFRJ also helped. But simply, there are 2 copies of the gene, and if Douglas knocks them out individually, we see no effect on the growth. If we do the double knockout, the parasites don't grow very well, and you can see here they have a distinctly altered morphology here in the knockout line. And actually, when we test the sensitivity against the chalcone, that knockout line shows reduced sensitivity or greater resistance, as if the target has been depleted, it's no longer there. So that toxicity associated with this target. Again, there's still some reasonable activity, which suggests that chalcone has polypharmacology. That's going to be a consistent theme that comes through. So all that work was done in the pro-mastigote parasite. What about in the amastigote form? What Douglas was able to do was to show that actually, if you look at the amastigote and the parent uninfected macrophage, in the amastigote, we can still detect cytosolic TX-MPX. We don't see any labeling of anything in the macrophage. That would be consistent with that selectivity index we saw there. And again, here, you can now see the intramacrophage amastigote activity. In that 22, the parent chalcone, there it is, in the dihydro form there. More importantly, the selectivity index. This one is relatively toxic. This one is not so bad. So how do we think it's working? So a very simple experiment. What Douglas was able to do is he took a population of parasites and you treat with NAT22. And what you can see is the number of parasites, viable parasites, decrease with concentration in a concentration-dependent manner. If you take those parasites out, you take a fixed number of those parasites, and then you assess for the amount of reactive oxygen species in there using a dihydrofluorocene analog that gets oxidized, what you can see is actually, as the number of parasites fall, the level of ROS in the existing parasites is going up. So there's an accumulation of ROS. If we do exactly the same experiments with the dihydroenone, we see very little effect. And this is probably associated with just general cytotoxicity at higher concentrations. So that's in the pro-mastigote again. What about in the intramacrophage amastigote? So again, if we do the same assay, we measure for ROS. We measure for ROS in an infected macrophage, and we add the chalcone in there, the amount of ROS goes up. If we add the dihydrochalcone, we see no effect. The amount of ROS stays the same in an infected macrophage. But in the uninfected macrophage, i.e. when the parasite isn't there, we see no effect of this compound. So this increase in ROS is due to ROS production in the amastigote. So our current hypothesis, the chalcone here is taken up by the macrophage, taken up by the amastigote in the macrophage. This is inhibiting its ability to deal with the enhanced ROS, the stress induced by the macrophage, and we get parasite death, selective parasite death. It's a working hypothesis, but that's where we are at the moment. So what we have now is we have a protein and we have a small molecule that we know binds in that protein. I don't think the chalcone will ever make it as an oral drug. Bartiera's got some very exciting evidence that shows it works in intralesional form where we don't have to deliver it. It's not very soluble. And I think if we try and give it orally, it will have too many other effects, too many other systemic effects. But it's a good tool to start building on new structures. And what we're doing now is doing some computational studies where we're docking this into computational models. And hopefully we've got a collaboration with Ashish Arora in Lucknow, who's doing crystal structure and solving and docking of the compound into the real structure. And that may help us understand better, and we can design better inhibitors of that enzyme. Okay, so that was the phenotypic type approach. I'm going to swap gears now. I'm going to go and show you a very brief story of a target-based approach. And the target-based approach we're going to do is based around targets in sphingolipid synthesis, so not a well-explored area of drug discovery. One of the reasons why it's not well explored is the pathway is highly conserved in most eukaryotic cells. This is sphingolipid synthesis in a mammalian cell, but it's very highly conserved down these first two steps to ceramide. So it starts at palmityl-CoA and L-serine, catalyzed by serine palmityl transferase to make 3-ketosphingolene. And that's then processed down to this pivotal intermediate, ceramide. And ceramide can be converted into a number of important signaling molecules, sphingosine 1-phosphate and ceramide 1-phosphate, and other bits of lipid metabolism. It can also be converted into sphingomyelin, the complex sphingolipid found in the plasma membrane. And it's this enzyme here that we developed to target. And those who've been coming to the earlier talks in this series will remember Paul Denny talking a bit about developing an assay for this process. And it's the same assay that we've used in this project here. So if we look at this in a bit more structural terms, why do we think this is potentially a drug target? So in chemical terms, so the pathway down to dihydrosphingosine, now we can compare plants, fungi, trypanosomatids, parasites, and mammals. In plants and fungi, dihydrosphingosine is converted to phytoceramide. So it has this hydroxy group at this position here. And this is taken up, converted by the enzyme inositol phosphoryl ceramide synthase, or IPCS, and reacts with phosphatidylate inositol to form IPC, so this molecule down here. In mammals and the parasites, we make ceramide. And in mammals, ceramide is converted by the enzyme sphingomyelin synthase with using phosphocholine as a second substrate to make sphingomyelin, whereas in parasites, we have the orthologous enzymes we saw in plants and fungi, IPCS, that's phosphatidylate inositol to make IPC. So the real take-home message in this slide is that the host and the parasite have different enzymes with different structural requirements. So what we wondered, or we speculated a long time ago now, is that if we can selectively inhibit this, could we get a selective drug for leishmania, or even all the parasites? So why do we think that? When we look at the enzyme, what we'll see is the enzyme has an active site histidine in it, and that reacts with phosphatidylate inositol, transfers this phosphoinositol head group onto the histidine, which then reacts with ceramide to form IPC. So if we inhibit this enzyme, we're going to see an accumulation of ceramide. And ceramide is a marker for programmed cell death, apoptosis, whatever you get in an organism. Excuse me a second. Okay, so if we can inhibit that, we'll see a buildup of ceramide, and we'll hopefully get selective cell death in the parasite. Excuse me, technical issue here. Okay, so how do we test that? Well, the enzyme is a membrane-bound enzyme and difficult to work with. So Paul very cleverly developed a system where we could take a yeast strain which had the fungal enzyme in it, knocked out and replaced on a plasmid. By means of a plasmid shuffle, we can get a yeast strain that's complemented by the leishmania enzyme. We can take a microsomal extract of this, this mutant yeast that's complemented by the leishmania enzyme, and that has the activity of leishmania IPCS, and we can use that to run an assay. And again, Paul has described that very nicely a couple of weeks ago. And the simple assay we developed is we found that this fluorescently labeled ceramide is accepted by the IPC synthase, and you can use that microsomal extract, and that will form this fluorescent IPC product. And you can detect the difference of these on a TLC plate or a simple ion exchange media, and we can measure the relative amounts of product you get from a fixed amount of enzyme by separating out the ceramide from the product down here. We do various bits of biochemistry, but more importantly, we can run very simple assays. And so we took that assay, we were able to convert it into a 96 well plate format. The co-worker at the time, John, did this by hand. So he screened 1,200 compounds that LifeArc very generously gave us from their compound collection. It's what's called the NINS data set, National Institute for Neurological Diseases data set. It's a set of 1,200 bioactive compounds, all cellularly active bioactive compounds of random modes of action that work. They're all sorts of compounds. None of them are repurposed drugs. So basically, in theory, you could intercept this pathway a bit further down. 57 of those compounds showed 70% inhibition of that crude enzyme extract in this assay. We retested those in the standard pro-masticate assay model using Alamo Blue as a live dead sort of monitoring process. And after about 24 hours, we saw 4 compounds showed activity in that assay that are better than pentamidine. One of those compounds I showed you was one of the currently used drugs. Pentamidine is not really used anymore, but it has been used as a drug for treatment of leishmaniasis. These compounds showed higher activity than pentamidine, which happened to be in this assay. What I want to focus on the remainder of this talk is this compound here, which is clemastine fumarate, which had the highest activity in that assay for us. Now, clemastine fumarate is, or was, a widely used over-the-counter available antihistamine used for a variety of infections and allergic responses, allergic rhinitis in particular. You get it over the counter, so it's a safe drug. So clemastine fumarate is this here. So we then retest it against the IPCS in a proper assay. We saw 2.5 micromolar. And when we test it against an intramacrophage amasticate model done at the London School of Hygiene for us by Vanessa Yardley, we saw about a 4 micromolar activity. So this was quite promising. If we're going to get a drug, there are 30 different species of leishmania across the globe that cause the disease. It's got to work against multiple ones, a good drug. So we test it now against 2 old world forms of the disease and 2 new world forms of the disease, 2 that cause cutaneous leishmaniasis, 2 that cause visceral leishmaniasis. We see submicromolar activity against all of them, which is very exciting, very pleasing in the pro-mastigote form. And for most of the rest of the talk now, because Bartier is particularly interested in cutaneous leishmaniasis in Brazil, and Leishmania amazonensis is one of the major causes of cutaneous leishmaniasis in Brazil, we're going to focus on Leishmania amazonensis. So we haven't done much with the other forms of the disease at the moment. So that was pro-mastigote. Does it work in the Leishmania amazonensis amastigote? Very pleasingly, yes, it retains that activity. It's now about 400 nanomolar or 0.4 micromolar. Macrophage cytotoxicity is about 20 micromolar, so it's got a good selectivity index. Just to put it on perspective, we look at the pro-mastigotes in the same assay. Our positive control amphotericin B shows very similar activity as clemastine fumarate in our hands. Okay, so we have activity against the pro-mastigote and the amastigote. Is it actually working as we think it is? Okay, and again, we go back to Paul. Paul, some time ago, developed a mutant parasite in which the necessity for the first module in the sphingolipid biosynthetic pathway was knocked out. So the sphingolipid synthase activity is still present, but it's no longer essential for activity. And so a drug that works against that activity should have lower efficacy against the parasite, and that's exactly what we see. We don't see complete resistance, and again, that may suggest that clemastine has polypharmacology. It still has some activity here. If we add back this module of the sphingolipid biosynthesis and regenerate the active sphingolipid biosynthetic pathway in these parasites, we restore sensitivity. So this would suggest that some of the activity of clemastine is directly linked to sphingolipid biosynthesis, and that IPCS could well be part of it. If we're going to test activity, we should be able to look at changes in the sphingolipid profiles as we go through. And this work was done by Dubiel, a postdoc in Paul's group, who previously worked with Mike Barrett as an expert in metabolomics. So he was able to do this work for us. And so we gave him some clemastine and he measured various metabolites. In particular, he looked at the ceramide levels. And here the black shows how much ceramide is in there in a wild type cell, untreated. And when we add clemastine fumarate, what we see is the ceramide levels go right up. It's highly consistent with inhibition of this pathway, blocking of this step over here, and increase in the amount of ceramide in the cell. Dubiel was able to show this occurs. Other bits of the pathway were similarly affected too. So very nice bit of work. We also looked at the other side of the pathway, and this was work done by Terry Smith at St Andrews for us. what Terry was able to measure is the phospholipids, and he measured the ratio of phosphoinositol to inositol phosphorylceramide. And in the absence of clemistine, in this particular assay here, we saw the ratio is something about 60-40. When we add clemistine, it drops to 25-75. So we're seeing a reduction in IPC relative to PI. Again, entirely consistent with inhibition of this biochemical pathway. So we appear to have various bits of activity. Very recently, Paul has developed a knockout, a CRISPR knockout, which is consistent with this activity, but that's data hot off the press, and I'll leave Paul to talk about it. He's on the call later. What Adubel was able to show, again, some clever work, he was able to show that you can generate resistance to clemistine. Takes a few goes. It's not a very high level of resistance, possibly indicative of other modes of action. But if you look at the where the resistance is occurring, a lot of them occur in genes associated with sphingolipid synthesis, again, consistent with activity against sphingolipid biosynthesis pathway, of which IPCS is a key part. Importantly, clemistine exhibits no cross-resistance to existing drugs. So it appears to be working complementary. This is quite encouraging because one of the things we're looking at now is developing co-formulations with Bartira in Brazil. Okay, I've shown you it works in the pro-mastigote, in the amastigote. Does it work in an animal model? It's non-toxic, so we put it into an animal model in Brazil, and it's a bit challenging for us, but what we're able to show is that if we give clemistine IP or interlesionally, it shows activity comparable to an antimonial glucanthine. And you can see here, here's glucanthine, here's clemistine interlesionally, here's IP, very good effect. Disappointingly, when we tried to formulate it and deliver it orally, we saw very little effect. And that could be for very rapid metabolism in the mouse, or it could be we simply didn't get the formulation right. And we think the latter, because clemistine is known to make you drowsy when you take it, and mice are no different. And if we look at the mouse when we dose it interperitoneally, the mice don't walk as far, they're sleepy, they're drowsy. We see a reduction in their motility. We do the same measurement when we dose the samples, when we dose it orally, we saw no effect. So we think that failure for oral dosing is we simply didn't get the drug on board, either a formulation or it's been rapidly metabolized. Something we're working on and trying to work on it. So this indicative again that clemistine has this drowsiness, that clemistine has multiple modes of activity. And that's really well known about clemistine. It's one of the reasons why it's not used so much. So here's an assay that colleagues at LifeArc did for us. They took clemistine and they looked at various different receptors that clemistine binds to. And clemistine is the green bars here. And so what we have over here is the H1 receptor. That's the receptor that clemistine was designed to target. There it is, good activity. But it also affects the muscarinic receptors, other histamine receptors, the dopamine receptors, some of the serotonin receptors, and whatever else. So it's not a clean drug in that sense. I'll explain what the blue bars are in just a minute. Lost some slides. Okay. Yeah, sorry. Overclicking. So clemistine exhibits polypharmacology, and that's also known. It's that clemistine is known to have an effect on this P2X7 receptor found on the surface of the macrophage. Leishmania also stimulates the P2X7 receptor. When the receptor is stimulated, ATPs influx into the extracellular ATPs taken into the cell and causes an ion flux, which contributes to the parasite killing effect. So we speculated that clemistine might be stimulating this effect, and we could do this by simply take a wild type parasite and we had extracellular ATP, the efficacy of clemistine is enhanced. If we knock out that receptor, there's no difference when we add extracellular ATP. So this seems to suggest that clemistine is also aiding the stimulation or the immune response to respond to a Leishmania infection. So we think this is strong evidence, yes, that clemistine isn't just working by inhibiting the IPCS, it is doing that, but it's also having polypharmacology. So what we think at this stage is we have identified clemistine fumarate, it's got potential to be repurposed. Maybe we need to work out how to formulate it and deliver it. but it's working in multiple ways, and that may be a beneficial thing. That may account for why it shows lower resistance. So drug repurposing potentially is a potential here. And because it shows low cross resistance, we're going to try and co-formulate it. This is all work that's still ongoing. So this is a living story. Okay, white slide here, because what I've shown you largely so far has been parasite biology aided by small molecules. Just for the chemist in the audience, I'll just show you a bit of some of the things we've been doing chemistry-wise. So when we saw clemistine, we thought, well, we should make it. Well, actually, it has been made. Jonathan Clayton's group, originally at Manchester, now at Bristol, have made it. Jonathan did this to develop some very clever asymmetric synthesis. So he did these steps at the bottom where he starts with his carbamate, does a very complex anion rearrangement to make the stereochemical core of clemistine. Now, it just shows you the challenge of it. This is the only reported asymmetric synthesis of this molecule here. And it's made as a mixture of the two isomers. So that's the methyl group going back into the board and the methyl group sticking out, 84:16 ratio. John was able to combine this with this proline-derived alkyl chloride. And then this shows the challenge. This is a very hindered molecule. It was challenging to do the alkylation. When you do the alkylation, you get a rearrangement of this head group to form this azopane in quite significant quantities. And he was able to end up with a mixture of the two isomers from which he was able to isolate this particular enantiomer of clemistine. So not efficient and difficult. I asked Johnson if he had any, and he said, no, sorry, we're only able to make about 1 milligram, and that's all decomposed in our storage. So we didn't really fancy trying to repeat this. So we asked the first question is, how important is this methyl group, this difficult to introduce methyl group? So it's very easy to make one without a methyl group. We make the head group much the same way as Jonathan did. We take proline, convert this alcohol. You can make the alcohol by a simple Grignard addition to these aldehydes. So you can make lots of diaryl carbonyls that lack the methyl group. Using a bit of gold catalyzed Lewis acid chemistry, we can very efficiently make a whole range of analogues so we can change the substituents around these positions here. However, when we make this compound here as a mixture of isomers, this is racemic, it's a 50-50 mixture here. This is diastereomeric mixture. The selectivity, the activity of this compound is considerably lower than clemistine fumarate. So obviously the question is that the methyl group that's important, or is that the stereochemistry, or is it both? We can make the single enantiomer.
Professor Patrick, we can't hear you.
You can't hear me. Zoom. Is that better? Can you hear me now? Did anyone hear me?
Professor Patrick, now it's okay. You can continue, please.
Okay, thank you. I don't know where you lost me. So excuse me, repeating myself. Very quickly to summarize, you can make clemistine. It's difficult because of this quaternary stereochemistry here, this tertiary alcohol stereochemistry. We were able to make mixtures of diastereoisomers, but these have lower activity. That could be because we no longer have the methyl group or the stereochemistry is wrong. So we can do some asymmetric chemistry. We can selectively make these diaryl carbonyls efficiently, both enantiomeric forms. And we can do the same reaction that Klein did, and we can make single enantiomers of this compound here. We've now improved this alkylation chemistry, but at the time we did it, and we also suffer from the same problem that Klein did. You get this azopane derivative from this chemistry. So it's difficult to make. We make both stereoisomers. And now when we test them, we can go through. We made all the possible stereoisomers now. There's a small difference in getting the stereochemistry of this proline head group right. There's a slight advantage to having the stereochemistry here, as shown. But if we get the stereochemistry right in the lower half, so we make this particular enantiomer of normethyl clemistine, what we find is the compound has the same activity pretty much as clemistine fumarate. And it shows a very similar profile in the way it interacts with biological material. So for example, in that radioligand assay, This is the compound in blue bars here. Shows a very similar profile to clemastine fumarate. Okay, so using that sort of simple chemistry, using that racemate molecule, and then making enantiomeric pure versions when we needed to, we were able to test bits of structure-activity relationship around it. You pick up some interesting observations. For example, this chlorine appears to be lacks We make the one that lacks any substituent in there, so there's no stereochemical issue at their center. We lose activity. We put 2 fluorines in there, small molecules. We retain some activity, but not sufficient, not as much. If we make easier to make linkers here, so for example, we made amides and triazoles and alkenes, we can see activity against the microsomal enzyme extract. We don't see activity against the parasite. So what we're introducing here, the rigidity of those linkers put in there seems to prevent uptake. What I've shown here is that we NH compound, so this methyl group adds to activity. It's not essential, but if we take that methyl group, that nitrogen out, we lose activity. And that's pretty much true for every compound we've made that we think targets the IPCS. It needs that basic nitrogen somewhere for activity. It needs a lipophilic group to fit it in. And that probably relates to the fact that it sits in the membrane. Okay, so all these activities, we're never really able to improve or even equal clemastine fumarate, and it was difficult to make. So how can we simplify the synthesis? Well, we looked at the molecule and we thought, what happens if we just simply move that nitrogen from this position round to here? This may be easier to make. We may get away from the problem of that rearrangement chemistry. And yes, these are very easy to make compounds. We can take the alcohol, we use our gold catalyzed chemistry to make this corresponding bromide, and then we can simply react with a proline-derived head group. So we make this in 2 or 3 steps from readily available precursors. Okay, so we can do that. But when we measured the activity of these, we made both enantiomers here with the racemate, comparing it with this one here. Really very little difference in activity. A bit disappointingly, then we think about the linkage between the oxygen and the nitrogen. We have 3 carbons. Here we have 2. Is that critical? So we went away and made the 2, the 3, and the 4 carbon very nicely. But when we get the right distance between these 2 ends of group, we retain activity. We make it too long, activity starts tailing off again. So now we have reasonable activity. Now if we can correct the stereochemistry here, which we can simply do by we take that asymmetric alcohol, we now get this alkylation. We've learned how to do this. It works much better with this series than it does with the proline-derived series. We now get the alkylation reasonable yields. When we get the right stereochemistry and the right chain length, we get activity that's comparable in our assays with clemastine fumarate. So we have a very simple compound now, easy to make. It's a much better model for us now to build and look at other structure-activity relationships. We've got some derivatives which show slightly better activity, not hugely. We're still working on those. Okay, interesting thing about this compound, this analog, it's got a lower macrophage cytotoxicity. When we look at those radioligand displacement assays, it inhibits far fewer of them, and that may account for that lower off-target activity, retains activity against the H1 receptor, retains an activity against some of the others. A lot of the others, it's relatively inactive, so that was unexpected, but it's rather nice. It appears to be on target in the same way. So here's our TLC assay. Here's the IPC formed. Here's clemastine, no product. Here's the compound here, SR157. No activity. We look at that delta LCB2 mutant assay. We see a similar profile. Again, it works that way. Again, we don't see complete resistance, so it probably retains the polypharmacology. We haven't looked against the P2X7 receptor yet for this compound series. That remains reigning. And does it work in the mouse model? Yes, it's this bar here. So this is intralesionally, so it shows a very similar intralesional effect as clemastine fumarate. So it's almost certainly working in the same way. So that's really where these stories are. We're still working with this compound. We're particularly trying to formulate clemastine fumarate. And I say, that's not my expertise, but we're happy to contribute to the product we make, supply material for people. So I hope I haven't gone on too long. What I've tried to show you so far, or tried to show you today, is that natural products are potentially good sources for new leads. The challenge can be to link activity with a target. And that's sort of essentially if we're going to develop them. And activity-based chemical proteomic methods represents one way of doing that. I've shown you that for this, and what we suggest is that this tryparodoxin peroxidase is potentially a new mode of action for anti-leishmanials that merits some further work, and that's ongoing work in Durham and in Brazil. And the second part I've shown you in a sort of target-based approach that's sphingolipid synthase activities conserved in eukaryotes, has a common active site, common mechanism of action across those cells, but selectivity is possible. What I didn't say is that antifungal activity has also been shown for these things, this class of enzymes. So we're not the first in the field. It's good, but it's maybe a new way for anti-leishmanial modes of action. And the clemistine fumarate is potentially an exciting repurposed drug for the treatment of leishmaniasis. Activity is probably a combination of effects, but that's not a bad thing. It probably leads to lower levels of resistance. We're looking at co-formulation, we need to improve formulation, and we're looking at testing new, more selective, more active analogues. As I inferred at the beginning, I just get to do the nice bit of the job, which is to talk about the science. The people who do the hard work are the students and the postdocs on the project. So here's some of them. It's been a collaboration between Bartira in Brazil, Paul and myself in Durham. So these are some of the students who've worked across them. And what's really nice to say, it's a real collaboration. So for example, Bex and Douglas did a lot of the clemistine fumarate work. Douglas helped with the vivo, Bex did the chemistry and a lot of the biology. Natalia and Douglas did the chalcone work. Chris did some of the early clemistine fumarate work. What's really nice is Bex and Douglas both got degrees from the Federal University of Rio de Janeiro and from Durham. So here they are at graduation. You can see Natalia, who started the early chalcone work in Durham, well wrapped up, and they're looking possibly suntanned and happy in America, in Brazil, in Rio. So let me thank them. Let me thank the other people who did the work. So Dougal did the metabolomics and the resistance work, something that I'm completely incapable of doing. I great debt of thanks to him. Stephanie helped us with expression and purification of protein. This is the rest of the group working on other bits of anti-leishmanial work. Natalia, Douglas, and Ariely started with Bartira. We're dependent on these people for their support. Paul and Bartira keep me on the straight and narrow. These people funded us. I'd like to thank them and their attention. Let me just finish the picture of the group. So here they are. This is last year. This is this year. This was actually last Monday. We're up on Hadrian's Wall, which separates Durham, which was built to keep, the Scots out of England when the Romans were here. So so this is a variety of the group. We come from all over the place, from Brazil, from the UK, from Hong Kong, from China, from Argentina, from Wales. It's been great fun. Here's Natalia, sorry, here's Ariely from Bartira's group at the moment, visiting us last year. So I thank them. In case you're wondering where I am now, I'm in Durham. Here's Durham. Hadrian's Wall runs across here, so that's where we were last week. Durham's about 300 miles north of London. It's an ancient town, certainly in UK terms. The cathedral is built here as part of defensive fortification, surrounded by the river, with a castle at the other end of this promontory to help fight the Scots. We do actually get on well. Stephen's in the network will testify to that. Here it is in the other sense. I'm actually sitting up here in the chemistry department, which is tucked away just here. My office is about there. And it's a nice place to be. You literally might have been here if you read Harry Potter. That was, a bit of it was filmed in Durham Cathedral. And I'll leave you finally with the voice of our former Chancellor of the University. The head of the university was Bill Bryson, as he once wrote, "If you've never been to Durham, go at once. It's wonderful." I'm afraid to say I ride a bicycle, so it can't be my car. So on that note, I'd like to thank you all for logging in and watching. I hope I've been comprehensible. Thank you very much. I'll stop sharing so I can see people.
Thank you, Professor Steele, for such a wonderful talk. Very informative, certainly, with lots of beautiful chemical space inside. We have some questions in the chat box. So if you can have a look, or maybe I can read the questions for you.
That'd be helpful. Yes. Why don't you do that?
Yeah. So the first question is, how can we avoid drug-drug interaction effect during drug repurposing studies?
I think you can't. You have to look for them. You have to be careful of them. You have to compensate them. Sometimes they can be useful, and sometimes they're obviously going to be risky. It's no different to any other drug discovery thing. When we repurpose a drug, we have to be careful about other drug-drug interactions. That's a fair comment, I think. Yeah.
Yeah. Another question, apart from ROS, that is reactive oxygen species, and ceramide buildup, are there other biomarkers of leishmaniasis?
I presume this is in relation to sphingolipid synthesis inhibition. Well, we see, you see a deformed, if you can see the parasite in the promastiga, you see a change in morphology, a change in shape. When we dose it, the parasites don't move so well when we treat them. Are there internal cellular ones? We see other changes in the sphingolipid metabolites. We see other things. We haven't looked much further for other biomarkers. Again, it's something that we could do. It's a good question.
Okay, thank you. There is one more. What properties a protein should bear to classify them as a target in bacteria and fungi?
Sorry, I didn't catch that question. Is it somewhere down? Can you repeat the question, please? I didn't hear the question.
Okay, let me repeat it for you. What
Thank you.
properties a protein should bear to classify them as drug target in bacteria and fungi?
Well, the same in any organism, ideally. A drug target needs to be, you need to built in, it needs to be, get a molecule to it, it needs to be bind to something, it needs to be essential really for survival of the parasite in whatever organism, or survival of the infected species, whether it's a parasite, a fungi, or a bacterium. And you need to be able to inhibit it with a small molecule.
Okay. Okay, there is another. Are some of these compounds active against Trypsinoma curzii?
We have sent clemistine fumarate. It showed relatively modest activity against T. brucei in an assay there. We're waiting results. We've sent it again to be tested against T. brucei, but I don't think it shows significantly strong activity. My preliminary data there.
Okay, I think we have one last. This is, I would like to find out if you have any interest in studying repurposing of drugs for other entities like lymphatic filariasis, onchocerciasis, or soil transmitted helminths.
It depends which way. We can't do those assays, but we could. We just don't have the ability to test against any of those organisms here. I guess we can send you samples of the drugs we make, and you can test them, but we can't do that ourselves here. Okay. I think all these things have to be a collaboration in the end.
Okay, excellent. I think, yeah, that's it. With this, I would like to thank Professor Patrick Steele for exciting lecture. It's such an honor for you, for us, that you presented your talk and your science with us. Thank you so much. Thank you, everyone, for joining in. Thank you. Bye. You have a good day.
Thank you, everyone.